So the following question has to do with finding the probability of a specific genotype from the F2 generation, but this time we're going to have an epistatic gene working, and actually we're going to have duplicate recessive epistasis. Now, the question says that F2 plants segregate 9/16 to 7/16, and the fact that we see the number 16 right here tells us that we probably have two genes at play, and we'll call them big A, little a for the alleles for the first gene, and the alleles for the second gene will be big B, little b, right? And we'll make it as a dominant recessive relationship. Now, if just one colored plant, now notice how it says colored. If we're talking colored, then we're only speaking about the 9/16 ratio, right? We're ignoring the 7/16. So this is the group that we're looking at. If just one color plant from the F2 generation is chosen at random and selfed, what is the probability there'll be no segregation of the two phenotypes among its progeny? Now, when you say no segregation of the two phenotypes among its progeny, right, which is colored and colorless, that just means one thing, which is true breeding. That's what true breeding really is. That's just the definition of it, right? So we're asking, what's the probability that an F2 generation will be true breeding for the dominant phenotype? That's what it's saying. Now, to get our 9:7 ratio that we see over here, right, we have to have something called duplicate recessive epistasis. Now, how does that even work? Well, go back to our regular branch diagrams. Remember when we had our F1 generation that was pure heterozygote, right? When it was crossed, we had this branch diagram where it was 3/4 chance of dominant for A, and then 1/4 chance recessive for A, and then these branched out to be 3/4 and 1/4 dominant for B and recessive for B, right? And so from this branch diagram, we got the ratios 9, right, to 3 to 3 to 1. And this right here was dominant at A, right? We didn't know what the second allele was, and dominant at B. And this one was dominant at A, but recessive at B. And this one was recessive at A, dominant at B. And this was right here, recessive at both, right? Now notice the reason we see 7 is because all three of these genotypes over here have at least one of the genes being recessive, right? And so because at least one of them is recessive, they're having the same phenotype. And this is called duplicate recessive epistasis, where basically you have two different genes, which is duplicate, and when they're recessive, they basically become epistatic to the entire phenotype, and they cause the phenotype of colorless instead of colored, right? So that's why we see our 9:7 ratio. And so of the 9, which are colored, we know that we have to have this perfect F1 heterozygote to get this basically distribution. To get the 9:7 distribution, our F1 must have been big A, little a, big B, little b. That's how we get this branch diagram, right? And so it's asking what's the probability that we're homozygous dominant, right, for both genes. Now, we have three options in our Punnett squares when we did the Punnett squares for any of the genes, right? We had 4 options, but among the dominants, we only had 3 different options. And so, of those 3 different options, right, what is the probability that you're homozygous dominant? Well, it was 1/3. And the same goes for the B gene, right? You had 1/3 chance of being homozygous dominant there. And so 1/3 chance of being homozygous dominant for A times a 1/3 chance of being homozygous dominant for B, that gives us a 1/9 chance total of being true breeding, right? And true breeding for what phenotype? For being colored. And so that answers the question of what proportion of our F2 generation will have no segregation among its progeny.